Please join me in welcoming Dr. Shanks.
Thanks for the invitation to speak with you today. So what had actually happened was I graduated from Berkeley, and then all of my colleagues and friends went off to companies like Yahoo and Google and Apple, and I ended up going to medical school. So that's sort of where I went now. But after graduating from UCLA, I came up to UC San Francisco, and I finished an internal medicine residency and then infectious diseases fellowship. And what I really felt, though, after doing that was I wanted to do something that could kind of merge the research with the clinical medicine. I knew that I enjoyed seeing patients. The big issue was there's this big disconnect, I feel, between clinical medicine and research. In that you have research, but typically it can take 10, 20, 30 years before you actually can see kind of impact to patients. And that's why here at UCSF, there's really a big focus now on developing what we call translational technologies, developing kind of research tools and kind of research technologies that can be readily translatable directly for patient care. So both for clinical medicine as well for public health. So I'll talk to you today about two of these technologies. The first thing we'll be talking about is kind of the use of what we call microarrays, and I'll define them later. And then the use of unbiased next-generation sequencing. Like, how many of you have heard of personalized medicine, or that term, or genome sequencing? Yeah, probably all of you have. So what I'll be talking with you today is the same thing, the same technology, but I'll be talking to you on how you can use it to look at not at human sequences, but looking at pathogen sequences. Okay? And I'll basically also talk about how we can take these technologies and move them kind of into the clinic or into public health. So I'd like to start by just giving you a few disclosures. One is that I get research support from Diagnostics, specifically to do viral discovery. I have support from Amazon.com to develop a cloud computing pipeline, development for analyzing next-generation sequencing data, and I'm on the advisory board for a genomics company. So let's just start with a case report. I'm going to talk about a 14-year-old immunocompromised boy who basically had an undiagnosed critical illness. And this is very similar to the kind of case that we see at UCSF. So I am actually on the wards as an infectious diseases physician. Very often we see patients that are admitted with an unknown disease that's likely to be infectious. So it might be an encephalitis, it might be pneumonia, it might be fever, it might be sepsis. And all of these diseases, the big issue is that it's very difficult to try to diagnose many of these infections because despite modern medical testing, we can't diagnose a huge proportion of these types of infections. And we'll be talking about that. And then I'll talk about kind of what I call the tools of the micro hunter, specifically microarrays and then next generation sequencing. I'll go specifically into details about the various microarrays that we've developed. I'll also talk to you about bioinformatics computational pipeline for those of you that are kind of more technically or genomics inclined. And then finally we'll go into basically direct applications. One for investigating an unknown outbreak in Central Africa, and the second back to that case and how that case can be applied to patient care in general. So this is a 14-year-old boy. He has severe combined immunodeficiencies, and he's immunocompromised. He basically was immunocompromised from birth. He had 2 partially successful bone marrow transplants, both from his mother, and he responded, so he had a partial response, but he was still immunocompromised. And he was actually on a monthly intravenous immunoglobulin because he has hypo or low levels of gamma globulins. He presented in April of 2013 with 6 days of headache and a fever. He was discharged after 1 day after his fever and headache improved spontaneously. This is his social history. He has no sick contacts. He did have 3 pet cats at home. And then he had an interesting travel history. He said about a year ago, he had gone on a missionary trip to Puerto Rico. And at that time, he swam in the river and the ocean. Interestingly enough, another member of the trip, a fellow traveler, a friend of his, was hospitalized for 4 days with what was described as fever and bloody urine upon returning home. And then also, 1 month before he was admitted, he went to a beach vacation in Florida where he swam in a pool at a resort that hosted feral cats. And he actually noticed that he was swimming in the pool and the cats were actually drinking from the pool at the same time. So probably not a good idea, but this was his history. And the other interesting thing was that he has a series of very unusual events that occurred kind of before his hospitalization. This was his trip in August. He then developed fever, headache, and conjunctivitis, which is an inflammation of the eye. It was treated with steroids and got better. And then he developed kind of more inflammation, what we call uveitis, which is kind of a deeper inflammation of the eye. He first developed it on his left eye, and then 1 week later on his right eye. And he was treated again with steroids and eye drops, and he got better. But then during this period, he then developed essentially these petechiae, which are blood spots on his skin. And that's actually a sign of bleeding. And then it turned out that he had very low platelets, and he was diagnosed with this disease, autoimmune disease called immune thrombocytopenic purpura. And that's also treated with steroids. So he was given steroids and he got better. Then he went on the trip to Florida, and then this was his first hospitalization. He was discharged after 1 day after the first hospitalization, but then he came back at the end of the month. And this time he was a little sicker. He had fever, he had photophobia, which basically is kind of eye pain, especially with respect to bright light. He had daily headaches, abdominal pain, and fatigue. He had a normal white blood cell count, which is a measure of inflammation or potential infection. But he did have an elevated erythrocyte sedimentation rate. That's a measure that we use for determining whether or not the patient is potentially infected or has an inflammatory process going on. And then they did a lumbar puncture, what we know as called a spinal tap. This is the CSF, which is cerebral spinal fluid. And I can tell you that it's not normal. Okay, the normal value should be 0. You should have no cells, no white blood cells, and no red blood cells. He had 125 white blood cells. Okay, so that's a high level. Basically, he has an inflammation in his CSF. He also had abnormal protein and glucose in his CSF. He had an MRI scan of his brain as well that was normal. And then he had extensive infectious disease workup. I won't go into kind of the testing, but he had over 100 tests that were sent, negative for everything. He wasn't given any antibiotics, and you might ask, why wasn't he given antibiotics? Well, they had a very strong suspicion because all the testing was negative. He had responded to steroids before. He had a strong suspicion that this was an autoimmune process. Or if it wasn't an autoimmune process, it may have been some unusual viral infection for which there isn't a good treatment. So he was not given antibiotics, and he improved spontaneously with the discharge from the hospital. And unfortunately, he comes back again. He actually was out of the hospital for about 5 to 6 weeks, and he actually went back to school. But then he came back, and this time he was very sick. Fever, headache, weakness, nausea, vomiting. We gave it 2 doses of rituximab, which is a kind of a monoclonal antibody that's anti-inflammatory. It's possible for possible autoimmune disease. And his cerebral spinal fluid still showed the pleocytosis, the white blood cells in his CSF. Except this time he had an abnormal MRI scan. And he had developed these patchy hyperintensities in the basal ganglia, which are parts of the deep brain. He had also developed in the interim what we describe as a basilar leptomeningitis. So this is what it looks like on CT scans. So this is called a corona section. So you're looking basically at the brain. Imagine you're looking at the head from the top. And you're looking here, so this is basically, these are kind of the ventricles and the cisterns of the brain. And what's abnormal are these, you see these kind of white spots there? That's abnormal. It should all be relatively gray, okay, uniform. This is basically thought to be either an infection or some sort of inflammatory process that's going on deep in his brain. And then this is also abnormal. So you see here, there's a very thin white layer here that appears to be enhancing, that appears to line the basilar cisterns. And this is basically, this is where the cerebrospinal fluid flows. And this is also abnormal. And this is basically a sign of basilar, which is the base of the brain, meningitis. So this is a basilar leptomeningitis because it involves kind of leptomeninges, which are here at the base of the brain. You can also see here, this is around the 4th ventricle. You can kind of see this kind of enhancement. This is abnormal. Okay? And along with headache, fever, he has a meningitis or an unknown meningitis or encephalitis. Okay. So unfortunately, he continued to do worse, he had no diagnosis. He ended up getting a brain biopsy, so basically stick a needle into the right frontal lobe where it's infected, and it showed that he had an inflamed meningitis, as we expected. But there was a granulomatous infiltrate, and what that basically means, there are a lot of inflammatory cells in that tissue. They looked at it for fungi, bacteria, and viruses, and found nothing, and he was finally diagnosed with a disease called neurosarcoidosis. Has anyone heard of that? Probably not. It's a rare disease, it's an autoimmune disease. But at least in this context, it's thought to be a disease of exclusion. It's basically a diagnosis that neurologists and infectious disease people give you if they don't know what's going on. Okay? And that's really what it is. Because like you see many patients with neurosarcoidosis. If it really is neurosarcoidosis, it would respond to steroids. Okay? Because it's really a classical autoimmune disease. So he was started on steroids. They also gave him this pegylated ADA, which is basically a medication that boosts his immune system. We recall he's immunocompromised, has severe combined deficiency, but continued to do worse. altered mental status. He then developed swelling in the brain, new onset Chiu seizures, and they couldn't control his seizures, so then they placed him in a medically induced coma. And they also placed a drain, and they actually started some antibiotics, but not to treat the infection, because they don't know what to treat with, but they started to prevent against infection of the drain. Okay. Any questions so far? I mean, this is a 14-year-old boy, basically. So perfect. He was healthy. He's immunocompromised, but he was healthy. He was perfectly healthy just a month ago, but now he's critically ill. Okay. And this gives you some idea of the studies that were sent out. Okay. And now infectious diseases, we go by what we call a differential diagnosis, just like in anything in medicine. You examine the patient, you look at the laboratories, you look at the imaging, you then formulate a list of what you think are the most likely possibilities. You would then send off these tests, so send off say anywhere from 5 to 10 tests. If those tests turn up negative, then you have to go back to the history, maybe try to find a little more about the presentation, think of more unusual etiologies, and then send off more and more tests. And this is just a partial list of his tests, and if you recall that he was actually admitted 3 times, so each of these tests were repeated at least 3 times, okay, usually more, basically. Okay. And all of the tests were negative. So what do we do? Well, I mean, I can tell you that there's a large percentage of patients who are like this, maybe not as critically ill as him, but are where we strongly suspect infection, but we don't know how to diagnose it. And indeed, this is a big conundrum in infectious diseases. Up to 25% of the time, we can't diagnose pneumonia. So any patient in the ICU, 25% of the time, we can't determine what the diagnosis is, even though it's very likely to be infectious. 50% of diarrheal disease is undiagnosed. Encephalitis, which is in his case the meningitis, up to 80% of the time, unknown cause. And even in well-described syndromes, you might expect hemorrhagic fever, like a hemorrhagic fever outbreak. You know, it may be sort of it may be kind of astonishing to you, but about 20% of the time, you have a hemorrhagic fever that looks like an outbreak, and yet we still don't know what causes it. Okay. So, and in addition, there's also been kind of the rise of what we call new pathogens, and these are pathogens like MERS coronavirus. Have you heard of MERS, or Middle East Respiratory Syndrome coronavirus? So this was in Saudi Arabia. It's now thought that potentially camels are the host. So, and so there have been some cases, very high mortality, 50% of people infected with MERS will die, and these are healthy people. You know, H1N1 influenza in 2009, which turned out not to be that serious, but H5N1 and H7N9 are both very fatal infections that very rapidly fatal bird flu infections. And then even non-viral causes like E. coli O104H4. Has anyone heard of that? Yeah. Do you remember what it was?
Wasn't that the German outbreak?
Yes. And do you remember how it was spread?
The sprouts?
Yes. Great. And then do you remember what was the typical patient in this outbreak?
I remember it being different than some of the other recall.
Yeah, it was. It's highly pathogenic. It had a high, in addition to giving diarrhea, it gave you kidney failure or renal failure, very high percentage, and the mortality rate was about 20%. So if you imagine, so something you ate can kill you, basically, a 20% chance of killing you is pretty severe. Interestingly enough, the main patient, the predominant patient in this outbreak actually were young women, young or middle-aged women, and it's been hypothesized that they were eating salads to stay healthy, basically. Basically, this was a bacteria that was spread with raw bean sprouts in salads. Okay, so the big question is, why can't we diagnose more infections? Well, the paradigm in infectious diseases is one bug, one test, okay? I want to look for influenza, I set up a PCR. I want to look for, I want to look for E. coli, for instance, I send off a stool culture, okay? Now the problem is that pathogens themselves, or any organisms that cause infectious diseases, can be very different. They can be what we call novel. In particular, this actually applies very well to viruses. What I mean by novel typically, I mean that these viruses are highly divergent. Their DNA sequence can be highly variable and can differ significantly from any other sequence in reference databases. So even if you sequence, say, a truly novel virus, you may not be able to identify it, or you may not even be able to classify it, or know that it exists. And this is a big problem. Now, the nice thing are that bacteria, fungi, and parasites are different because they have very large genomes. And because of the size of their genomes, you should be able to identify them. They're very likely to have some resemblance to something else in the database so that you can at least identify them and determine how much of a threat they may pose. In addition, a big issue is that the existing tests have very low sensitivity in detecting organisms that are present at very low concentrations. Okay, I want to go over viruses as a good example. These are current viral detection methods. You can grow it, you can actually stain it, stain viral proteins, you can actually look at it under an electron microscope, or you can do PCR, polymerase chain reaction, to amplify the genome, or serology to look for antibodies. That's about it. Okay, now they have their limitations. Probably these are rather laborious. They take a long time to culture. They can take up to weeks to culture. DFA is kind of insensitive. EM is just not very specific. I mean, you can look at these and like, does anyone know what these are basically by looking at it? Anyone that's worked with viruses before? It actually turns out that it's pretty classic because you can see, kind of see this halo formation around the virus. It is a specific virus. Any ideas? So it's a coronavirus. It's SARS. So that's SARS, basically, yeah. But you can see that it's a challenge to try to identify it just by looking at what it looks, just looking at its shape. Serology and PCR, these are the mainstays for viral diagnosis. The problem is these suffer from the one target, one bug paradigm, meaning that you're only looking for one or a few agents at a time. You're not looking for everything. In addition, as I mentioned before, viruses are extremely diverse. This was a study that was published in MBio. It was by the Lipkin lab at Columbia University. And they actually mathematically estimated that there are over 320,000 mammalian viruses that exist in nature that have yet to be discovered. And that's actually thought to be a severe underestimate. I would say there's probably 10 times that amount probably exists in nature. This was kind of a lower limit of what has yet to be discovered. So the good news is it doesn't look like infectious diseases people will be out of a job anytime soon. Okay? In addition, so the general idea of looking for what we call a pan-diagnostic is, why not look at DNA? Why? Well, it turns out that genomics, or the study of DNA, including the genomes of microbes, is a fantastic way to identify pathogens. And that's because nearly all microbes that we know about are made of DNA. Okay, so the idea is just by looking at the DNA sequence, either through microarrays or sequencing, in potentially you could detect bacteria, all of them, viruses, all of them, parasites, all of them, fungi, all of them. You're only going to miss kind of one type of organism, or you probably shouldn't call it an organism. You're only missing one type of pathogen. Does anyone know what that is? Right. Yes, both of you said it. Great. So they're both prions, okay? And why? Well, because they're a protein, okay? So they're protein. Prion's the etiologic agent of mad cow disease, of Creutzfeldt-Jakob disease, with neurodegenerative diseases in humans, okay? But basically, you can detect everything else. And these are the tools that we're using. So one's called microarrays, and the other's called next generation sequencing. And specifically for next generation sequencing, we are doing what we call unbiased next generation sequencing. So what that means is that we're not targeting anything in particular. We're just sequencing everything. I call it sequencing the hell out of a clinical sample and then trying to identify what's in it. Okay? And this permits us to cast what we call a wide net. So instead of a kind of a single fishing pole, we're aiming to add kind of a huge fish net and capture the full diversity of potential pathogens with a single test. Okay? So it sort of began with the development of what we call the ViroChip. Now, who has heard of the ViroChip? Those of you who work with pathogens. So I was actually a postdoc in Joseph Derisi's laboratory. He was one of the pioneers in developing this technology. And the general idea of microarrays is they're simply basically spots of DNA that are basically placed onto glass slides or some other substrate. And the idea is that based on the pattern of the spots, in principle, you should be able to identify pathogens. So we have what we call the ViroChip, which basically represents all of the 2,000 viruses in the GenBank database. So you might imagine we basically synthesize DNA spots, we print them out, and in principle, based on the pattern, you can identify any of 2,000 viruses. Okay? That's a general idea. We also deliberately use very longer probes that are used in other microarrays. So we have basically probes that are 70 nucleotides in length. And that's important because then you can identify mismatches. They might have, you could have, as I mentioned before, you have the viral divergence problem. Viruses are highly variable. Well, we can sort of compensate for that somewhat because we have very long probes that can pick up divergent viruses. To give you an example, this was actually originally used to detect SARS coronavirus. And at that time, SARS was an unknown coronavirus. However, we did have probes to known coronaviruses, and the SARS coronavirus did cross-hybridize to those probes. that enabled us actually to identify a potential novel coronavirus in a clinical sample from a patient. Okay, it's also been used to diagnose respiratory and diarrheal infections. I've been able to show that it has pretty good sensitivity and specificity relative to gold standard PCR. We've also designed written programs that can do kind of automated interpretation, so you don't have to look and manually look at every single spot. The program will just spit out an answer for you. And the latest version, it costs about $100. It's printed on a commercial Agile platform. Okay? So this gives you an idea. These are two cases. These are blood samples. These are both patients from India who actually developed a hemorrhagic disease. And on the right are basically two patients from the United States who have acute liver failure. But the idea here is that you can readily diagnose. You can just see clearly that this is chikungunya virus. This is dengue virus type 1. You notice that we can also distinguish between type 1 and type 2. Hepatitis A, hepatitis B. And this is called a cluster map. What I'm showing you here are basically the probes to different types of influenza viruses. And these are basically 20 of the original samples. So these actually include the original samples from Mexico in 2009. So some of the original samples during the pandemic 2009 H1N1 influenza outbreak. And then these samples from BC, called BC, they're from British Columbia. They were actually returning travelers from Mexico. So this was basically in April of 2009 when the pandemic first occurred. And you can see here that when we look at it with the viral chip, it's pretty clear that there's a concentration not of seasonal human H3N2, which was the most common strain, not human H1N1, but actually swine H1N1. And the idea behind this is that if you look at this pattern, you'll say, let's say you got a nasal swab from an infected patient. Well, you could quickly just look at that, and visually you can say that this is not a typical seasonal influenza. This is potentially a novel influenza. And it turned out that it actually was. So the idea is that even though we didn't have the sequence, or even though we didn't know about H1N1, you can still be able to identify a novel pathogen because it maintains conserved probes. Okay. Any questions on that? No? Okay. We're sort of extending it right now. We've actually developed what we call the flu chip. We're adding basically probes specifically to do influenza subtyping. These are provided by the St. Jude Reference Center by Dr. Richard Webby, who's kind of a World Health Organization expert in flu. And he's provided us these are all of the known human and avian strains at the time. And you can see here that we have pretty good accuracy, about 96% accuracy in being able to subtype flu strains. And the plan right now is actually to take this and we're doing it to do surveillance in animals and humans. So we're currently doing some studies. We're looking at flu strains from Africa, and we're also looking at those from the United States, from Iowa, actually, and we're trying to survey swine. The idea is basically by surveying, say, pigs, where we know these influenza viruses may be coming from, we may be able to basically detect them before they have a chance to jump over to humans. Okay. We're also doing work with Lyme disease, and again, so I'm not going to focus on all of the work. I'm just specifically going to talk about kind of our detection, which is the use of the microarray, and then we'll go into sequencing a little bit later. Oops. Okay. And so what we've done is we took the viral chip, and then we decided, well, why not detect more than just viruses? Why don't we add kind of targets to detect really all bloodborne pathogens? So our idea here is we want to pick up all known bloodborne bacterial pathogens, Borrelia burgdorferi, that causes Lyme disease, Anaplasma, Phagocytophilum, Rickettsia, Bartonella, Tularemia, Leptospira, protozoans like Babesia, Microdia duncani, Toxoplasma, Candida cryptococcus, which are fungal pathogens in the hospital. And then we want to maintain kind of all the viruses we want to look for. And the idea is, can we do this in kind of a single assay? But this is a little tricky. And the reason it's a little tricky is viruses, there are lots of viruses in clinical samples. So typically, and usually we get a sensitivity of about 100 copies, so we can detect 100 viruses. That's not a big problem because, say in the setting of influenza, you can have titers of 10 to the 5th up to 10 to the 9th. Okay? Now for hepatitis B, for instance, a patient who's acutely affected with hepatitis B, they can actually have viral loads of 10 to the 11th, 10 to the 11th copies. So you might imagine that's actually fairly easy to detect. The problem with these bacteria are, especially for bacteria like Borrelia burgdorferi that causes Lyme disease, when Borrelia is in the blood, it's actually in blood at very, very transiently, and it can be in blood at very low titers. So we actually need to go down, we need to do better than 100 copies. We need to go down to maybe 10 copies, 1 copy, or even kind of using kind of stochastic considerations like 0.1 copy per mil. So our idea with this is that we're adding probes, but we want to maintain, we want to keep it as sensitive as possible and actually make it more sensitive than what the viral chip was. So the viral chip we were getting down to maybe 100 to 1,000 copies. We want to get down to like 1 to 10 copies. So how do we actually do this? Well, we tried using the same strategy with the viral chip. And the idea was to just amplify everything, put it on the probe and microarray and see what's our sensitivity. Turned out we got the same sensitivity. We got sensitivity of 100 copies to 1,000 copies. So that didn't seem to work. So what we did instead was we basically designed primers and actually targeted a highly conserved region among all bacteria. It's called the 16S ribosomal RNA. So what we did is we specifically designed high efficiency probes to that. And you can see here, these are 42 Babesia probes. And we were able to show through like kind of spiked dilutions that we are now getting down to a sensitivity of like 1 genome. So we can actually detect 1 genome. I can take Babesia, for instance, of the culture, spike it in to negative whole blood at a concentration of 1 genome per mil, and I can detect it. Okay. So we've achieved kind of like the limits of detection that you really want, like down to 1 copy. Okay. This shows you the same thing with Borrelia. So this is again a kind of what we call a cluster map. So what you see here, these are all of the probes. These are the different samples. And what you can see here is that, when you see red, it basically means that the probe lights up. Okay. So if you look at 10 to the 4th, everything lights up. We can definitely detect 10 to the 4th. 10 to the 3 is the same. Even 10 to the 2, we have very, very good detection. By the time you get to 10 to the 1, you may lose a little bit here, but you can still detect it. But what's amazing is that even at 1, we can still detect it. If you kind of squint your eyes, you can actually see there's a very thin band, even though there's probes are stronger here. There's a very thin band here. And we were actually able to show that we could detect PCR positive samples that are near the limits of detection for PCR. So these are serum samples from patients with acute Lyme disease, and they were taken directly from Martha's Vineyard. So they're basically a Lyme endemic area. You can see here that these samples have an estimated titer of 1 to 10 copies, and you can see here that we can detect, Borrelia with high sensitivity. Okay. So currently, this is kind of what we're doing with the validation. We're basically picking what are arguably the 3 most important tick-borne pathogens, bacterial pathogens and parasitic pathogens, Borrelia, Babesia, and Anaplasma, as well as negative controls. And we're trying to achieve kind of a sensitivity down to 1 copy with 100% specificity. Yes?
So in the clinic, do you care about sort of the load or maybe concentration of bacteria in the blood, or just the presence or lack of?
Yeah, that's a good question. With bacteria, the thinking has been that we don't know the significance of the actual load. Okay. Yeah. Now it is true for viruses, though. It's clinically significant for many viruses. So, for instance, hepatitis C, HIV, hepatitis B, those are CMV, EBV. These are viruses which cause disease for which the viral load is important. But for bacteria, really the goal for clinicians, they just want to know, is it there or not? Can I diagnose it or not? Is it positive or negative? Any other questions? Okay, so this gives you an idea. So these are actually the probes that you saw here. You can see here, they very nicely map to kind of the 16S gene. So the idea is that we're basically targeting a very highly conserved region within the Borrelia genome. And by doing so, we can kind of get the sensitivity that we want, okay? There have been there's some caveats to actually looking for 16S. Some people say that this is great. This is actually the principle behind all microbiome work. So if you think about microbiome, pretty much what they're doing is just sequencing 16S and characterizing bacterial populations. Now for detection, it appears to work really well. The one caveat is that even though you may detect it, you can see that it doesn't do a great job of distinguishing between exactly what species it is. So if you do want to know what species it is, you might want to use a different test or target additional regions. It turns out clinically, nobody, right now we don't really know the significance. We don't know whether you get infected with Borrelia miyamotoi, Borrelia burgdorferi, Borrelia zelli. We don't know. Clinically, they all present similar as Lyme disease. So they're treated the same way. Okay, I want to move into a new technique. This is called next generation sequencing. Okay, the general idea is, instead of using a microarray, what we're doing here is we're truly using an unbiased technology. We're simply amplifying everything in a clinical sample, and then we're using analysis of the sequence to reconstruct genomes or reconstruct sequences of potential pathogens. One big issue is that this is required development of both new specimen preparation protocols and new tools. Although now the capacity is pretty amazing. We can get down to like 1 billion sequences routinely. I have 2 MySeqs in my lab. We can generate like 50 million sequences in about anywhere from 6 to 24 hours. Okay, starting from the sequencing library. This is what I call a needle in a haystack problem. This is a serum sample from a patient from Africa who is infected with HIV. You can see here the vast majority of reads are human, and then you have a very, very small fraction of virus. But you can see that those are enough. The 842 reads out of 10.5 million reads are enough for us to detect HIV, adenoviruses, GBV, and Hep B. So basically 4 viruses detected using a single assay. Okay? And what's also made this attractive is now that they're kind of portable instruments. I know that this is kind of a point of care kind of diagnostics group. And the nice thing is that now these instruments they're not quite point of care, but at least they're portable. Okay? So to give you an idea, the MySeq is only about maybe 3 feet by 3 feet. And you're welcome if you want to drop by my lab to just take a look at it. It's basically it's a simple instrument. You can kind of move it around. This 454 Junior is even smaller. The Ion Torrent is smaller, and these are about. This is 125K. This is coming down. It's like 75K now. This is about 100K. This one's still a large machine, but these instruments are getting smaller and smaller and cheaper and cheaper. Okay. And to give you some idea, I've been collaborating with the Biosafety Level 4 laboratory, Eric Corral's laboratory in Gabon, Africa, and he has a field MySeq. So we placed the MySeq basically, and it's an area where basically power is not even reliable, and yet they've been able to do a few runs basically right in the heart of the Congo. So this becomes kind of potentially a portable point of care type of instrument. Okay, and it has enormous capacity. You can generate up to 100 million reads in under 24 hours. Okay, so what can you do with this? Well, one big issue is you have 100 million reads. The big question is how are you going to analyze that data? Okay, and this is a big problem. So for those of you that know Moore's law, okay, this is basically Moore's law that's kind of graphed. What you're seeing here is Moore's law is linear if you're considering kind of a logarithmic scale. Okay, however, you can see that deep sequencing is like roughly an exponential graph on a logarithmic scale. So we're basically the capacity of our computational capacity is basically being outstripped by the amount of sequencing data being produced. Okay, and practically, this is the bottleneck in that we basically have our 100 million reads. The main challenge is to get rid of the human background reads. We don't care about the human reads. We're not doing cancer sequencing or genome sequencing. We want to identify the small subset of pathogen-specific reads. So specifically to do that, we have to identify all human reads. Then we have to align those reads to everything in GenBank. Okay. Now the human database is about, or the human genome is about 4 gigabases in size. GenBank is 52 gigabases in size, and it's actually doubling every 2 years. Okay, so this is a big problem. We have an enormous amount of data. How can we rapidly identify pathogens? So traditionally, this has been a computational bottleneck. Okay, and originally I thought one way to actually deal with this, one solution is maybe you want to make your computational power scalable. And the idea is expand it beyond the context of kind of my single research laboratory. Move it on to the UCB QB3 computational cluster. This cluster has about 5,000 nodes on it, so 5,000 processors. The cloud computing, you can actually do Amazon EC2, can get you up to 10,000 cores simultaneously to do your analysis. And then ultimately, you might make a very nice web server, and the idea is that you can kind of access and analyze your data from your iPhone or your Android. Okay. Now, how does this work in practice? Well, on a single machine, to analyze 100 million sequences takes 3 months. Okay, so if you're thinking about a rapid clinical diagnostic, that's not going to work too well. Patients will be dead by then. Okay. Now, on the other hand, network 18 days, still a little better. Cluster 24 hours, and cloud 6 hours. So you're thinking, well, cloud is fantastic. I can recruit up to 5,000 cores and kind of get it analyzed very quickly. But then it becomes a problem is that you have to pay. Okay. But the good thing is that I actually have a grant. Okay, but that grant is not. Basically we ended up kind of using up the grant in like 2 weeks. Okay. So then I asked Amazon for more money. I want more time on this. But the problem is that it boils down to be about $400 to analyze. And this is just the analysis. So this has nothing to do with the sequencing or the cost to actually prepare your library. It costs $400, and that's not practical. So what I did was I actually teamed up with a group here at UC Berkeley. Who's actually in computer science here? Any computer science people? Who knows, like, So I've been teaming up with the kind of Ian Stoica, Richard Karp, I don't know if you know, they're computer scientists here at Berkeley. So I've been teaming up with them and also some people at Microsoft. And what we've done is we basically designed a new algorithm to do alignment very quick. So the traditional alignment is called BLAST. Because for those of you who've done BLAST alignment, BLAST was actually published in 1986, okay? And actually it still remains, in terms of biology, it's the #1 most cited biological article. It's the BLAST, it's that original 1980 article. But if you think about over the past 30 years, there haven't been any improvements to BLAST. People are still using BLAST. And what do we use BLAST for? Well, we use BLAST to align sequences to a reference database, okay? Now, so what we've done is effectively we've replaced BLAST with another algorithm called SNAP. And SNAP has a benefit that it's about 1,000 to 10,000 times faster than BLAST, okay? And the idea is that you can kind of implement the same sort of pipeline, but instead of taking 3 months, this is a pipeline that can be done in 20 minutes, okay? So how does that work? Well, one thing is that SIRP, which is the name of our pipeline, which uses SNAP, is very, very fast. This gives you an idea. This is basically number of reads that you need to analyze. These are different algorithms. Probably VOTI and VWA are commonly used in human genome sequencing. This is BLAST. You see BLAST takes 19 hours to analyze 1.25 million reads. SNAP can do that in 40 seconds. And when you get to kind of typical counts, like 25 to 125 million reads, SNAP does pretty good. We're talking 7 minutes, 2 to 7 minutes. Whereas for BLAST, it would take anywhere from 16 to 80 days. And I don't even want to talk about 1.25 billion reads, where SNAP can do it in a little more than an hour and a half, and BLAST takes 2.2 years. Okay, that's on the exact same server. Okay, same server, same processor. So basically, we've dealt with that bottleneck. We have a pipeline now that can actually analyze that data in 10 minutes to anywhere to 15 hours. So it's still within the ballpark of where you consider you can use this clinically. Okay. And the cloud computing costs, as you might imagine, are less now. So now we're down to like $4. Okay. A couple of experiments that we did is one, we wanted to see what is the sensitivity of this strategy. So this is where we basically took HIV, we spiked it into whole blood at different concentrations. And from 3 million shotgun sequences, what we call 3 million randomly generated sequences, you can get a sensitivity of 10 copies of virus. So you can get a sensitivity equivalent to PCR just by generating 3 million sequences, at least for detection of HIV. Okay, we'll be able to show that this is true for other viruses as well. Okay, so we have what we call an NGS clinical workflow, and this is a workflow that we're actually doing now routinely in the microbiology lab, which is a CLIA-certified facility. But the idea is that we can, with a total turnaround time of 12 hours to 2 days, we can take any sort of sample, serum, whole blood, respiratory secretions, cerebrospinal fluid, stool tissue, bone marrow. We can actually generate data, within 2 days. Okay. So I'm going to give you like 2 applications of it. So the first is acute hemorrhagic fever in Africa. Well, it turned out that Africa, in the African Congo, there was an outbreak of hemorrhagic fever. So what is hemorrhagic fever? It's a disease that's usually caused by viruses. It occurs in tropical climates. It's commonly transmitted to humans by arthropods, like bites of mosquito or tick or rodents, characterized by high fever, petechiae, these kind of red spots that you see from internal bleeding in the skin, internal bleeding, which can be basically manifest as vomiting blood or have blood in the stool, and then low blood pressure and shock. And this was an inaccessible village, so the only way to get to the village actually is to fly in or ride a motorcycle. Okay. Does anyone know the hemorrhagic fever viruses that circulate in Africa? Any ideas? Ebola, absolutely. Ebola is one of them. Any others? Marburg, Ebola and Marburg. Those are classic filoviruses. Anything else? Dengue? Yes. Dengue is a hemorrhagic fever syndrome. Anything else?
There's one called Mancala or something like that? Or
There's Mocala. Yeah, that's pretty rare. But it is a hemorrhagic fever virus. Yeah. Anything else? Hanta does cause hemorrhagic fever. It's not commonly found in Africa. Hanta causes hemorrhagic fever, but more often in South America, Latin America. It's a very common cause. Yeah, but there's one in West Africa that I'm missing, which is a lot. There's like 400,000 people actually get it a year. It's much more than Ebola. Anyone know the virus? Arenavirus? Lassa fever virus. Okay, so Lassa. Okay, so you guys did a great job. You guys actually went through all of the categories. So these are the 4 known viral families that cause hemorrhagic fever. We have Arenaviridae, you know, Bunya viridae, well, Rena viridae, Lassa probably is the most well-known. Bunya viridae, Hanta, Filoviridae, Ebola and Marburg, and Flavy, which, you know, dengue and yellow fever. Okay, so you guys went through all of that. And these are the 4 viral families that are known to cause acute hemorrhagic fever. Okay? And these are how they circulate. So basically, we were investigating outbreak located here in kind of the Congo, or kind of the west end of the Congo. And you can see here that what circulates here are really Ebola and Marburg. This is a filovirus endemic area. If you look at where Lassa primarily concentrated in West Africa, Crimea and Congo hemorrhagic fever, which is a Bunya virus, is kind of scattered throughout Africa. And there's a new virus, a new virus called Lujo virus, that actually is in these 2 places. It was basically somebody in South Africa got infected and traveled here and basically infected someone else. So it was a very small kind of outbreak, basically, of hemorrhagic fever. So this was our hemorrhagic fever. It was a very small outbreak, occurred in a remote village called Mangala. And when it happened, there was a 15-year-old boy, a schoolboy, who came in with, did not have a fever, but had hemorrhagic symptoms. So bleeding from his eyes, bleeding from his mouth, hemorrhagic and vomiting and diarrhea. And he died in 2 days. They had their health center, you know, they really did not have modern medical care. They really didn't care. This was a remote area. And then basically, a week later then, a 13-year-old schoolgirl, who wasn't a classmate of the boy. There were no known contact between the boy and the girl. She came in with high fever and hemorrhagic symptoms, and she died in 3 days. Okay? And then a week later then, the school teacher, the male school teacher of the boy and girl, also developed fever and hemorrhagic symptoms. And at that time, they realized that this is potentially like an outbreak. And they actually flew him to the closest city, which is the city of Boma. He got modern medical ICU care, and he survived kind of the hemorrhagic fever outbreak. So what we did is we took the serum from the last person, from the male teacher, and we looked at it with unbiased next generation sequencing. We also looked at it with the microarray. We didn't see anything in the microarray. And I'll show you why we didn't see anything in the microarray later. Now you might ask, you know, why wasn't it tested for Ebola, Marburg? It actually was. It was actually tested for all 4 of these hemorrhagic fever families, and it was negative. Okay, so this is what we did. We basically initially we used kind of a 454 pyro sequencing, and we got a single read. Okay, and so then we decided, well, you know, 454 pyro sequencing, you can generate maybe about 50,000 sequences. We decided we would decide to throw the book at it. So instead of 50,000 sequences, we generated 140 million sequences. Okay, we did all from the same serum sample, and out of it, we were able to basically start with that single read. And by de novo assembly, you can actually assemble the entire genome within 10 cycles. So this was done within about 2 hours. We were able to get the entire genome. So we not only detected something, but we sequenced the entire genome of this novel virus. And this is a virus that we're calling the Congo virus. And what's interesting is that the Congo virus is a rhabdovirus. Okay, who knows what rhabdoviruses are? Anyone heard of? Can anyone give me the name of the most famous rhabdovirus? It's a Lissa virus, carried by bats.
Causes rabies?
Yes, rabies. So the rabies virus is a prototype rhabdovirus. But if you actually look at rhabdovirus, you know, rhabdovirus is not one of these families, okay? So that was sort of unusual, okay? Well, you know, this is, and the other thing that was really weird was that this is a very divergent virus. Now, rhabdoviruses, rabies is the best known example. It infects more than 100, there are more than 175 viruses that infect a wide variety of hosts, vertebrates, insects, plants, humans, and has not been previously linked to hemorrhagic fever. And this is called a phylogenetic map. And you can see basically based on the deepness of the branch, it's just how divergent, how different it is. This is actually a virus that kind of extends at a very early, kind of branches out at a very early stage. Branches out and has a very deep root. And that suggests that it's a highly divergent virus. In fact, this virus is so divergent, it only has 30% amino acid identity. It has like close to 0% nucleotide identity. So this is something that would not have been detected by microarrays, would not have been detected by PCR. In fact, we really needed next generation sequencing. It was really the only way. And even then, we were only able to identify a few reads that had some homology. The closest relative that was sequenced is called Tigro-Gargan virus. No one's probably ever heard of it. I hadn't actually heard of it either. Tigro-Gargan virus is a virus that infects cows. So it's actually found, it was described in cows in Australia. They get this disease, like fever in cows, and it's spread by biting flies that basically bite the cows and give them disease. Now, is there precedent for rhabdoviruses causing hemorrhagic disease? And indeed there is. If you look at this phylogenetic map, these are the mammals, these are the bats, rabies is here, these are the plants, and these are the fish. Fish developed this disease called viral hemorrhagic septicemia. It's actually very well known among fishermen, where basically they'll have spontaneous bleeding. So there is precedent for hemorrhagic disease from rhabdoviruses. And that's why it kind of gave us an initial clue that this may be the etiologic agent. In addition, it was kind of intriguing that the closest relative to this, even though it's a deep branch, the closest relative to this is a virus that basically bites cows and causes fever in cows. So again, so we think that this is potentially a hemorrhagic fever agent. So what we did was we generated, and the nice thing about the sequence is once you have the sequence, you can do a lot with it. So you can reconstruct the entire virus. You can actually synthesize the virus. So I'm actually doing that right now. We're trying to synthesize the virus. We're not trying to synthesize in San Francisco, so don't worry about that. We're doing it in a biosafety level 4 laboratory, maybe in Texas, okay? But we are trying to synthesize the virus. But beyond that, what we're doing, what you can do is you have the sequence so then you can express proteins from the virus. And that's really useful because once you have proteins, you can then interrogate people for what we call serological response. We can look for antibodies. And we were able to show, actually, these are 5 people or 6 people from the Mangala village. This is actually the patient who had acute hemorrhagic fever. You can see here that basically this curve shows that he had a very strong antibody response. So he was feeling infected with that virus. He actually had a copy, he had 10 million copies of the virus in his blood at the time that he was sick. But interestingly enough, there's actually the nurse, that nurse who actually took him, the fellow nurse that took him to the city of Boma where he got modern medical care, that nurse was also incidentally infected, although that nurse did not exhibit any hemorrhagic symptoms. And so is there precedent for people getting infected but not having hemorrhagic fever? Well, there are cases for certain viruses. So a good example is Lassa. So Lassa virus, for instance, only 20% of the time if you get infected will you develop hemorrhagic fever. About 80% of the time you either have no symptoms or you have very mild symptoms. So there is precedence for hemorrhagic fever viruses not causing hemorrhagic fever. It's not really the case with Ebola. It's thought that 80% of the time if you get Ebola, you'll get hemorrhagic fever. So if you need to get infected with hemorrhagic fever virus, you'd rather get infected with Lassa rather than Ebola. That's the take-home message. But again, I don't want to get infected with either. Okay. But this was a nurse who was asymptomatic. You can see here that they have antibody studies. The other thing is that we're now doing a large kind of antibody surveillance for this virus. So the question is, where does Bacongo reside? Where did it come from? And that question we haven't answered yet. But we have been able to show that we can detect a prevalence of about 10% in patients specifically in Bacongo and in closely the Republic of Congo. So this appears to be a virus that's predominantly in Central Africa. When we screened every place else in Africa, we couldn't find it. So it suggests that people are being exposed. More recently, though, we have a report that we've actually found some sequences that are similar to this virus in bats. So I don't know if this is going to turn out to be like Ebola in Marburg, where it originates from bats, or like SARS, for instance, which originated from bats. But that's sort of where we are with this virus. Okay? And this is kind of the model that we have, but we believe that these 2 patients died, these kids died, and then followed by the nurse who got hemorrhagic fever but survived, and then passed it on, and it sort of ended here. But this is a pattern that you typically see with Ebola. It comes from the community and then spreads among healthcare workers. Okay, let's go back to the case, okay? So we've already called, we have this 14-year-old boy, critically ill, okay? So this is called a clinical laboratory workflow. We got the sample, this is when he was in ICU, we got the sample on the 21st of August, okay? Within 48 hours, basically we had a diagnosis, so we just ran it through. And what I want to show here is that this shows the relative time it took to do the library generation, to validate the library, to actually do the sequencing. This is the analysis. It used to be the analysis would go on, for weeks and months, but now we've managed to bring it down to 97 minutes. So the idea is that this will fit within a workflow, and we decided to kind of use a conservative specimen prep, so that's why it took 48 hours. But now we could probably do the same thing in maybe 16 to 24 hours. Okay. Yes.
Earlier, you showed a pretty large panel of tests. Do you know how long all of those tests were taken just to compare this with?
Oh, how long it takes?
Yeah.
Each test has a turnaround time usually between 8 hours to like 4 days. So typically, yeah. So it sort of fits within the test. But usually these tests are ordered serially, so that's why it took so long. That's why the patient was actually in the hospital for a month before we got the sample. Yeah. Yeah. So and when we added up the cost of those tests, it was over. The tests alone were about $50,000, but the cost of his stay was more than $400,000 basically, because of, you might imagine how all those procedures you know, icy stay is expensive, you probably know. I mean, that was a cost, but, you know, they should send us a sample immediately, but yeah, they didn't. So, yes, that was okay, fine. Okay, so what we found out was that this is actually a case of leptospirosis, okay? We actually found a very clean sample. This is cerebrospinal fluid. These are the actual sequence reads mapped to the 2 chromosomes of Leptospira board-petersoni. Now, it turns out it's not actually Leptospira board-petersoni. It actually turns out to be a different strain. But what is lepto? Well, leptospirosis actually is the most widespread zoonosis in the world. It occurs basically when you get infected by contact with contaminated water and soil. And in fact, how many of you, do any of you triathlons or are really into sports, basically, or triathlons? Well, it turns out that this is well known as causing outbreaks among triathlons or people that go whitewater rafting. Because you get exposed to the water. You basically get exposed to contaminated urine in the water. At least here in the United States, it's usually caused by runoff from cows. So cows are infected with lepto, and the cow urine, eroding urine, kind of runs off into the lakes. Okay, and then basically so there was actually a large water triathlon. 74 out of 640 athletes became sick with lepto, acutely sick with leptospirosis. And this just by participating in the triathlon. And there are outbreaks among whitewater rafters as well. And it has many protein manifestations. The clinical manifestations are so different that it's very hard to diagnose on the basis of just a clinical presentation. It can cause meningitis, hepatitis, nephritis, infection of kidneys, rash. It can cause kind of sepsis or septicemia. It can actually cause conjunctivitis, and it can also cause uveitis and thrombocytopenia. Does that remind you of anyone's case? So this is, in hindsight, actually it was sort of interesting. No one had actually put this case together. He had seen like 5 different physicians. And it turns out that his medical chart was sort of scattered. And so when we actually put it together, so I'm presenting it to you the whole case together. When I presented this to resident report at UCSF just last week, the residents got the diagnosis within 5 minutes. Okay, and it really just shows you kind of the important. The fact is, it's not just a technology. It really takes clinical acumen to some extent cannot be replaced by technology. So it shows you they actually got the diagnosis. That was the number one thing of the differential. So I was actually quite impressed. So basically, it turns out that this is a strain that's kind of rare. It's Santaricide. The most common strain of leptospirosis is called interrogans. That's a worldwide strain. Santaricide is only found in certain places: Puerto Rico, Caribbean, Latin America, and Taiwan. Okay, those are the only places that's been found. It actually hasn't been found anywhere else in the world. And what was interesting was that we subsequently tried to confirm this. It turns out that all of the clinically validated assays, including one that was done by the Centers for Disease Control, so Dr. Renee Galloway is the CDC expert on lepto. She did her CLIA-validated assay and it was negative. Okay, and why was it negative? Well, it turns out that they validated the assay on common strains of lepto, and they did not validate it on Santaricide, which was this one strain. And it turns out, I mean, we actually think it's just a crummy PCR, basically. But anyway, this is the CDC's PCR, okay? They're clinically validated PCR. And what she had to do is she actually had to use like a new mix. So she had to convert it from a clinical assay to a research assay. And if you do so here, you can now see that it's now positive. This is just positive control. But you see here, this amplification shown in red, that's actually the positive result for the leptospirosis. The hallmark, the gold standard for diagnosis is a serological test. It's looking for antibody to lepto, but there was a problem with this patient. Does anyone know why? Yeah, well, he has severe combined immune deficiency. He doesn't make antibody. And in fact, when we tested with antibody, it was negative. So this is a good example of one where clearly next generation sequencing is another example where next generation sequencing was necessary to actually make this diagnosis. So even if the residents, for instance, at UCSF, if they had thought of lepto, they would have sent off an antibody test that would turn out to be negative, and they wouldn't have made the diagnosis, unfortunately. So, but it's a good example of kind of the power of sequencing. So what had actually happened, it turned out that we discontinued the steroids. We started IV penicillin, which is the treatment of choice, is the gold standard treatment. He had a dramatic clinical response. He actually improved within 12 hours. His seizures improved. He was out of the hospital in 2 weeks, basically. This is from being critically ill in the ICU. And it was actually thought that he got exposed during a missionary trip to Puerto Rico. In hindsight, his fellow traveler fever, nephritis, and very likely had lepto. So we actually went back, tracked down the fellow traveler, and tested him. He's not immunocompromised, his friend. So we tested him, and it turns out that he was antibody positive to lepto, basically. So they had both been infected with lepto in Puerto Rico. Okay. It wasn't just lepto. This gives you some examples. I'm working with a large number of institutions on this. Africa, Stanford, University of Washington, California Department of Public Health. These are just give you some examples of the various pathogens that we've discovered in clinical samples. Again, these are all done with next generation sequencing. So we're not using any specific primers. We're not targeting anything. We're just taking the sample and looking at it. You can see here a wide variety of different pathogens. So the one thing is, since this is a kind of point-of-care diagnostics talk, one of the quite big questions is, though, even though you have a powerful diagnostic, you always have to think, kind of, if you want to kind of market it or look for commercial applications, you have to think, like, what is the market for it? And then similarly, what is the clinical impact? Can you develop results that are actionable? And when we're thinking about that, we feel that there's some areas of like need. So areas of clinical need, what are these kind of respiratory, diarrheal, immunocompromised fever panels for detection of viruses? Because we don't have good tests for viruses in general. Another is for kind of being able to identify, not diagnosing bacterial infections necessarily, but being able to identify positive cultures. Because the problem right now is that we can grow something like bacteria, but it can take like, in some cases, several days to actually identify what bacteria it is. And one nice thing about culture is it's already amplified. So it's very easy to take that stuff, put it on sequencing, and you can probably identify within hours. You should be able to identify exactly what you cultured up. We're also interested in looking at what we call the politically hot diseases like Lyme disease, where we might be able to get kind of both interest, not only interest from institutions, but also interest from the public, basically, because it's thought to be kind of a big threat, at least in the United States, because we just simply have inadequate diagnostics for Lyme. So that's another area that we've been focused on, Lyme and Lyme-like illness. So I just want to summarize just a few points. One is that I think these are very promising technologies. Power of it lies in that we're looking for multiple pathogens simultaneously. And now that we're developing, and we, as well as a large number of groups, are developing new pipelines that can really improve both computational turnaround time and have the ability to make these accessible to hospital and clinical laboratories. The big issue right now has been the bioinformatics issue. But my thoughts are that once we develop user-friendly software, web-accessible software, potentially, hopefully under kind of a HIPAA protection for patient confidentiality and privacy, we would be able to make these available to hospitals that don't have the bioinformatics expertise to analyze the data. So really what physicians are looking for is kind of a black box. They just want an answer. They don't actually want to know how you arrive at the answer. They want an answer that's accurate. Okay. So I just want to thank the people in my lab. Eric Samoya is a clinical scientist in the laboratory. Svia Nikash is a postdoc who developed the pipeline. Jerome and Deanna have been working on the TICK project. Scott is also a bioinformaticist. Steve Miller is a close collaborator that's been working with the clinical microbiology laboratory. This group in Predict have been working with hemorrhagic fever in Africa, and then I also work with the California Department of Public Health. And I'd like to acknowledge kind of funding from the NIH, Abbott, Amazon, and the UC Discovery Grant for encephalitis. Thank you very much. I'll be happy to take questions. Any questions?
So, I mean, with timeline, you can think of when we become routine procedure.
Yeah, if you asked me a year ago, I would say it's still like 5 to 10 years. But with the rate at which improvements are being, at the rate at which new instruments are coming out, and the rate at which there's been interest in validating it, I would probably say like 2 to 3 years is reasonable for it to start getting used. We're developing, for instance, NGS. We're validating it now in the clinical laboratory, and our thoughts are to develop data that we could actually use for our FDA application. And I can tell you that just last month, the first next generation sequencing assay, it's not infectious diseases, but it's for diagnosis of cystic fibrosis, has been approved. So on that instrument, the MySeq sequencing instrument, you can do, it's been FDA approved. You can do a clinical assay for diagnosis of cystic fibrosis. I don't think that infectious diseases is going to lag that far behind. Yeah, but it's a really good question. I would say 2 to 3 years.
How much are we talking about? How much cost?
Cost, that's a great question. The cost is still a little high. So what we've been able to mitigate the cost somewhat by barcoding, by doing multiple samples per run. But to give you an idea, each run, if you had to add the cost of reagents, and not including labor, is about $1,500. And we can barcode usually like 6 or 7. So we can bring it down maybe to as low as like 200, 250. 250, I would say. That's not including like labor. You would still need a technician to actually run the assay. But it's not that cheap. That's not that expensive, basically. It's clearly, if you consider, like, for instance, the reimbursable cost for an influenza PCR is $350. I don't know if you knew that, basically. So it's getting there where it might be competitive with the existing tests. And obviously, it clearly has the advantage that you're looking for a large number of targets rather than a single target.
But at the same time, from the bioinformatics point of view, you need large sequence data for different class of pathogens. Virus, fungi, you know.
Well, no, the idea is that we're generating sequences and we don't care. We're not targeting viruses or fungi. I think what you mean is that we do need large reference databases. We do need large reference databases, and we do need computational capacity, yes. Yes, but the idea right now is that we're moving it all to the cloud and using these rapid algorithms. So for instance, you can take 100 million sequences and you can now upload it. In fact, if you want to test it out, you can download it right now, basically. It's not publicly available, but I can make it publicly available. You can download it, you can upload it to the cloud, you can run it and analyze it, and it'll spit out all of the pathogens, bacteria, fungi, viruses, and it'll do it for like $5. It'll do it for like $5, yeah.
Which are sequenced already.
Yeah, well, you generated 100 million shotgun sequences. You just don't know what they are.
Right.
Yeah. But it can identify, like all of those identifications were made solely by shotgun sequencing. So we're not targeting any individual
No, I
pathogen.
understand. What I mean is you are identifying by comparing existing sequence.
Yes, that's correct.
So
Yes.
if there's a certain area, not too many, for example, you know, I think there's a lot of fungi sequence now available.
Yes.
Protists, not that many. So if any protist-related disease, you may have a problem.
Well, I actually don't think we're going to miss it. The reason I don't think we're going to miss it is because the actual, the amount of protists, bacteria, and fungi that are actually human pathogens is actually a very small part of kind of the diversity exists within animals. And the idea is there's pretty good sequencing coverage of known human pathogens. I agree with you, if there's a new protist, or a new fungus that has no homology, that suddenly comes out, causes an outbreak or something, this would be hard to detect. It's only as good as your databases. But now what people are doing is the databases they don't just include human pathogens. You know, something like GenBank NT, it really has all of the environmental sequences, all the sequences that we're getting from, you know, surveillance of insects, animals. We know a lot of these novel or kind of outbreak pathogens are coming from reservoirs, animal reservoirs. So the idea is that we would be able to, as we get more and more sequence information, as we do more genomic surveillance, we would actually constantly be improving our databases. Yeah, but you are right in that if a pathogen comes out of nowhere and is highly divergent, we would not be able to detect it. Yeah, it would be very difficult to detect with that. Solely by that. Yes.
If someone was co-infected with 2 influenza viruses,
Yes.
how is it going to interpret that? I mean, are this?
We actually had that case already. We can, it can detect both of them. And the way it does it is that it detects some reads that are in common to the 2 influenza viruses, but then it detects some reads, if you have a good enough coverage, it detects some reads that are specific for one subtype or another. So for instance, we had a patient that was infected both with influenza A and B at the same time. And for that, we had some reads that were specific to flu B and some reads that were specific to flu A. So it is...
Would that work for, say, H7N9 and H1N1 also?
Yeah, it would work. It would work. But the odds of being co-infected by 2 influenza is actually very low. I would say the odds are greater that you get co-infected by, say, like a virus and a bacterium or 2 different viruses. Yeah, it's probably not that common. That wouldn't be a problem for sequencing, assuming you have enough coverage. That may be a problem for the microarray for various reasons, because the microarray is based on conserved sequences. So in some cases, it's difficult to detect co-infections with the array.
So, for example, if the new virus, which actually comes from assortment of 2, you will see the sequence of some of this and some of that.
Well, it depends on your coverage. If you have good enough coverage, for instance, you would be able to what we call do de novo assembly. You look for overlaps, and eventually you can stitch together the whole genome. And then you would look at the whole genome, and then you could say, hey, this is a new strain because it was a reassortment of this influenza. So that's a general idea. If you don't have enough coverage, then that's a problem. Yeah.
What-
If you only
What's
have a
the
few
what's the
reads.
depth in your 2-day, 200, and 2,000?
Oh, yeah, well, so it depends on how much you're multiplexing. So each run now, let's say the MySeq is about 50 million sequences. And if you multiplex, let's say 5-way, you can generate about 10 million reads per sample. Okay. Now, out of the 10 million, 99.99% of it's going to be human. Then you have a small fraction that hopefully is your pathogen. But you don't need a lot. I mean, if you detect, in theory, you only need like one sequence. But of course, then you have to worry about contamination or within the laboratory. But usually we can get anywhere from like 10 to several hundred reads, and that might be enough for you to assemble a genome, something small like a virus. You won't be able to assemble a bacterium, basically. Bacterium is about 4 megabases in size. So we're really using that to detect bacteria, not to assemble the genome. But we're hoping that for viruses, which have much smaller genomes, you have good enough coverage, you should be able to assemble the whole thing directly from the clinical sample. Yes?
Is there a difference between different type of NGS tools, like IronTorrent, MySeq? Is there any difference?
Yeah, all of them have their advantages and disadvantages. So the answer is yes, there are differences between them. And that's why I would say in terms of the ones that are probably at this point are probably considered the most practical, at least for what we're doing, it would be IronTorrent or Illumina, basically. Those are really the ones there's another company called Oxford Nanopore, I don't know if you've heard of that, but they reported 2 years ago that they have this essentially a portable, almost like a pocket type of sequencer where you store your data on kind of a USB stick. I haven't seen anything. I haven't seen an instrument yet. So they sort of presented 2 years ago. But their what they claimed was that you could actually use that for true point-of-care diagnostics. You can do point-of-care sequencing. Now, in practice, if you actually look at their technology, you're not going to be able to generate a lot of reads. You only generate about 10,000 reads, compared to say 50 million, which is what we get with Illumina. But it still will probably be pretty useful for specific applications. Any other questions?
Does read length has any implication?
Read length has, a lot of implication. I didn't go into that detail, but the idea is that you want it long enough so that you can unambiguously identify something. If you have very short read lengths, sometimes you can't tell whether it's a bacteria, it comes from bacteria, a human sequence, or maybe a virus. Nowadays, with these longer read lengths, so we're getting with the MySeq, we're getting up to 300 bases paired in, 300 base pair paired in. It's not a problem. But it was a problem early on where the sequences were only 50 bases long, or in some cases 35 bases long, then it became a problem. But now I think they've pretty much dealt it. Once you have a sequence longer than 100, it tends to be pretty specific for your target. Yeah, we have a different problem in that, for instance, let's say you want to diagnose a retrovirus, okay, like HIV, for instance. Well, the problem is retroviruses have stolen genes from the human genome. So you may get sequences to say oncogenes, and it may say that that's HIV, but it's not actually HIV, it's just an oncogene in the human genome. It's just that person's genome. So we're getting kind of issues with that. And for instance, bacteria, a lot of bacteria have borrowed, human-like genes, or they have genes that are similar to human proteins, and then it becomes difficult, unless you have good coverage, of really distinguishing that as a bacteria versus the host. So these are the problems that we're actually getting with respect to kind of identification.
So you throw away all the human sequences?
Yeah, well, we don't throw them away. We store them. We store all the sequences. So that's actually an area that I didn't talk about at all. But one area of my lab is really doing research intensely on doing host response and being able to diagnose disease by looking at the human host response. So we actually have all of that data. Right. Yeah. And the idea is that you could, for instance, look at the human host response, be able to use it for diagnosis or maybe prognosis, be able to determine clinical outcomes by looking at the human host response. So all the data is there, and really we just need like someone that's interested in mining it. So if anyone wants to do that, we have that data. I mean, we have more than right now we have more than like 10 billion sequences in my database. Yeah. And like 99% has not been analyzed because we don't care about human beings. We haven't been caring.
When you say host response from the expression level, RNA?
Yes, I'm talking about gene expression, like RNA.
As distinct though from the genome of the human.
Yes, yeah. I'm talking about RNA. Yeah, I'm talking about RNA. Yeah. It turns out that the method we use, for instance, the method that we use for detecting viruses is actually related to an RNA-seq method. So we're using the same method, we can get gene expression, mRNA levels, and at the same time find the virus. Yeah. So there are actually like 2 postdocs in my lab that are working on that problem right now. But you're right. Right now, all we're doing is storing it or ignoring it, basically. In fact, we're actually we're trying to look for ways of getting rid of the human sequence. So I have some ideas of depleting clinical samples because if you look at it, we really are wasting a lot of sequence. I'm throwing away 99.99% of my data. So I mean, if there's any way that you can just enrich maybe like 5, 10-fold, that you can see that it would be much more useful, make the assay that much more better.
As you're a medical doctor, and yet you're so good at this technology, computer stuff, how did you get so good at that? I don't expect that from
Oh,
medical
no, I
doctors.
didn't tell you that. I was an ECS major at Berkeley. So at that time, my friends went off to Yahoo and I went to medical school. Well, it's not. But I don't think that's how you get into it. It's just you need to have like a passion for it. No, what I quickly realized is that I didn't want to like program websites the rest of my life. I mean, it was sort of like I mean, true, you can make lots of money and get lots of stock options, but I mean, the question is like, what are your priorities? So for me, I really enjoy taking care of patients, but at the same time, I didn't want to be just a medical doctor. I felt that I felt that I could use some of my technical background to really be able to develop technology that can be helpful to like a wider group of the population level rather than single patients. Although I still see patients. I mean, I still do ID consults, for instance, in the hospital about like 1 to 2 months a year. So I still like that, and it's nice. It's nice being in my position, I think, because I have a good idea of like what's where the clinical need is, and how can you really translate the technology. I think that's been kind of the big barrier, and in fact, even at UCSF, it's a big challenge. Well, we have great researchers and we have great clinicians, but it's really hard to find that bridge and finding people that are willing or able to kind of translate kind of the research to really kind of bench to bedside type of work. Yeah, how that helps you basically. But like early on, even as a medical student, I basically programmed these. I actually did some of the original programs on like the Palm Pilot and developed like medical software and stuff like that. I like programming, so that's I never really programmed. So we're just doing the same thing. It's just for a different application. Yeah. Any other questions? Yeah, my email is charles.chiu@UCSF, so feel free to email me if you have any questions or want to visit the lab. Thanks.